Everyone, myself Ramya. I'm a biology teacher. Today I'm going to explain about blood groups of class 9th, chapter 3, animal tissue. So before getting into the topic, let us understand what exactly is a blood. So blood is the most important fluid in a human body. So what does a blood carry? It carries oxygen, it carries food, it also carries waste materials. So every human being have an same color of a blood, which is red color. But now there is a question: do you think everybody is having the same type of blood? Maybe no. For that, we have to understand things called antigens. So in our red blood cells, there are certain antigens. These antigens are nothing but proteins. They are on the surface of the blood. So based on these antigens, the blood has been divided into groups. That is called ABO system, where A, B, AB and O. We also have an Rh system where it tells whether the blood group is positive or negative. So let us get into an activity. Let us see how to identify the blood group. So the blood groups have been introduced by Mr. Karl Landsteiner. So he introduced about blood groups. That we have 4 different blood groups, that is A, B, AB and O. That O. To conduct an activity, we need certain solutions called A, B and Rh solutions. So what is the purpose of A solution? Anti-A solution helps us to find A group. Anti-B solution helps us to identify B group. Rh solution detects positive or negative blood groups. So the next topic is blood agglutination. What do you mean by blood agglutination? That means already mentioned that in our body, in the red blood cells, on the surface of the red blood cells, we have certain proteins. Those are also called as antigens. So in the previous slide, we have seen 3 solutions: anti-A, anti-B and Rh solutions. These also contain specific antigens in them. When the antigens present in a human body and these antigen solution mix up, we can detect the blood group. That is called blood agglutination. Yes. Now if you see this video, you can understand when a blood is inserted into a solution, an anti-A solution or anti-B solution, when you mix them properly, you can find the lumps forming here. This is called blood agglutination. That means anti-A solution when mixed up with a blood which is having anti-A or anti-A agents in them, when they mix up with each other, they stick to each other. Both the antigens present in the solution as well as blood, both mix up with each other. When they mix up with each other, the red blood cells lumps up with each other. So this tells us that which group of blood we belong to. Now how to find the blood groups? For that, as mentioned earlier in the class, that we need a glass plates, we need certain 3 glass plates, we need spirit, we need a needle and we need anti-solutions. So take 2 plates, in one add anti-A solution, in other one add anti-B solution. Now take a needle and pierce your finger and take some blood drops. Put in anti-A and anti-B solutions. After putting the blood, now take a toothpick and mix them properly. After mixing certain time, you will observe that in one particular plate, you see the blood get lumps over here. But when you observe in other plate, the blood doesn't get lumps, but it get dissolves into it. So as mentioned earlier, we were talking about blood agglutination, where when anti-A serum or anti-A solution has uh, anti-A solution as anti-A agents in them. So if in our blood we have anti-A antigens, these both mix up with each other and the lumps are formed, which indicates that we belong to blood group A. The same experiment repeats for B as well. If the lumps are formed in B, that means the person belongs to blood group B. If both the solutions A and B, you find lumps in them, that means the person belongs to a blood group AB. Talking about O. After mixing the solutions, you don't find any kind of lumps forming in either of the plates, which indicates that the person belongs to blood group O. So, blood groups have been completed. Now, let us talk about the positive and the negatives of the blood group. For this, take 3 glass slides. Each has anti-A solution, anti-B solution and RH solution in it. After adding the solutions, similarly take a needle, pierce your finger and add blood drops in each of them. After adding, take a toothpick and mix them. After mixing, you see the blood lumps in anti-A solution and RH solution. What is the purpose of an RH? RH will detect whether the blood is a positive or a negative. So, when a blood in an anti-A get lumps and RH solution also get lumps, that means the person is having A positive blood. Similarly, with B also. If a person's blood lumps up in B and RH solution also get lumps, so that means the person is having B positive blood. Similarly, we can find for AB and O also. This is how we find blood groups. Thank you.